Good afternoon. In this talk, I will describe some of our recent work on epileptic seizure detection and localization using multi-channel scalp EEG. Specifically, our methods blend classical techniques with recent advancements in deep learning. Our hybrid strategy allows us to overcome many of the unique challenges in this domain. Before diving into the methods, I would like to set the stage for much of the work that my lab does. So here are some rough statistics. By the end of this year, approximately 1.25 million children in the United States will be living with autism. 49,000 young adults will suffer their first schizophrenic break. There will be 795,000 incidents of stroke. 16.2 million people will go through a major depressive episode, and 5.5 million elderly adults will manifest the early signs of Alzheimer's disease. Now, I'm showing this slide just to give you a sense that neurological and psychiatric disorders are complex, and they're also pervasive, both in our society and also across the lifespan. The goal in my lab is to combine analytical techniques from artificial intelligence, signal processing, and statistics with multimodal data and unique clinically motivated insights to tackle the problems of biomarker discovery, therapeutic planning, and outcome assessment in clinical neuroscience. Now, clinical neuroscience is a field that inherits all of the challenges of high-dimensional data analysis and none of the advantages that have propelled advancements in domains such as computer vision or speech recognition. So specifically, a lot of the times we are grappling with small data sets, and this is for a variety of reasons. So first, the acquisitions can be very costly, and additionally, the patient recruitment. Even in scenarios where we are working with sort of a clinical workflow, the acquisition protocol can be inconsistent, and environmental confounds often make it difficult to fuse information across different protocols and different sites. Now, at the same time, we are also dealing with very complex phenomena, and as you'll see in the rest of the talk, oftentimes the differences and the distinctions we are trying to find are very subtle. In addition, there's massive patient heterogeneity, so these discriminative features can vary from person to person, and it can be very difficult to validate many of the methods and results because there is often a lack of ground truth information. Now, in fact, my lab is working on a range of problems that include predicting multidimensional clinical severity in autism, using genetic variations and trying to link them to cognitive dysfunction in schizophrenia, mapping the boundaries of eloquent cortex to improve brain tumor resections, and predicting the level of paralysis in spinal cord injury. Now, of course, as I mentioned earlier, this talk will focus on epilepsy. And here's a very brief outline for the remainder of the talk. I'm going to start with some background about the application as well as prior work in the field. So epilepsy is one of the most common neurological disorders. It affects roughly 1% of the world's population, and it's characterized by recurrent and unprovoked seizures. In addition to the seizures themselves, there are a great deal of psychological and physical comorbidities in epilepsy, which, when taken together, is partially responsible for the 5-fold increase in mortality. Now, the first line of treatment for epilepsy are anti-epileptic drugs, also known as AEDs. However, 20 to 40% of patients are considered medically refractory, meaning they continue to experience seizures even when they're taking AEDs. And for these patients, really, if we can determine that the seizures originate from a single focal onset in the brain, then the best course of action is to go in and surgically remove this onset. So as you can imagine, there are a great deal of challenges in trying to figure out what part of the brain is triggering seizures and where we should go in and resect. Now, scalp EEG is arguably the first and foremost diagnostic tool in medically refractory epilepsy. And here, as the figure illustrates, what would happen is a patient would come into the hospital and a set of electrodes are adhered to their scalp, usually in some predefined, we call them montages or organization. And they'll actually sit in the hospital in an epilepsy monitoring unit for between 2 and 7 days until we're able to capture a number of seizures in these EEG channels or signals. And really, once all of this EEG is recorded, the main goals for a clinician are seizure onset detection, so to identify when in time the seizures are occurring, as well as seizure localization, so to provide kind of a rough lobe, hemisphere, or sublobe type of location where the seizures might be originating. Now, despite decades of work on signal processing, machine learning type tools, it's actually quite striking that almost all of the diagnostics are manual. And by manual, what I mean is that an expert essentially would scan hours and hours of multi-channel EEG recordings like this one, and then go in and try to visually identify what are the signatures of the seizure and where it might be starting. And so this slide actually highlights why automated methods really struggle in this domain. And specifically, if we look at this image, which is fairly typical of an EEG recording, the most salient features in this image, so the high amplitude or very strange and uneven oscillations, they're actually artifact. Right? So one is muscle artifact and the other is eye movements or eye blinks in this case. And it turns out the seizure signature can be very, very subtle. And so here, what we see is the seizure is marked by kind of this rhythmicity or rhythmic oscillation, which is much, much lower in amplitude than the noise. And sort of complicating matters even further, these seizure signatures can vary both in their shape and type as well as where, in terms of which channels they appear in. And so this is kind of why the problem domain is quite challenging. Now, with that said, automated seizure detection has been an active area of research for the past 3 decades, and it's really the most common application of signal processing and machine learning tools in epilepsy. So automated methods are roughly organized into this 4-stage pipeline shown on the slide. So first, the raw EEG signal is pre-processed using a combination of filters. And so a low-pass filter is used to remove muscle artifacts or other types of artifacts from the signal. High-pass filtering is used to remove DC trends and very slow drifts. Oftentimes, a notch filter is applied as well, and this will remove the influence of the power line. And finally, the EEG signal is windowed into short 1 to 2 second intervals, which provide approximately stationary signal characteristics. Now, the bulk of prior work focuses on feature selection, where the goal is to isolate ictal or seizure-related signatures. And so common themes in feature selection include Fourier domain analysis, wavelet filter banks to capture certain shapes, and nonlinear entropy-based features as well. And then finally, the last step is to classify each time window as either baseline or seizure, thus providing temporal information about the seizure onset and offset. So prior work has explored both conventional machine learning classifiers as well as end-to-end deep neural networks. But what's pretty striking is despite the wealth of research, these detection pipelines are not robust to patient heterogeneity, and as a result, they've made limited impact on the standard clinical workflow. Now, seizure localization using scalp EEG takes a different route, and it focuses almost entirely on trying to improve the EEG spatial resolution by deconvolving the EEG signals, which could be sort of 10 to 40 across the scalp, onto a set of current dipoles or distributed sources at the millimeter scale in the brain. Now, unfortunately, these inverse solvers require careful annotations about the seizure interval, which actually there can be disagreement across experts, so it might not be consistent. And more importantly, inverse solvers are sensitive to things like physiological noise, the number of EEG channels, and the underlying head model. As a result, these inverse solvers are not considered reliable enough for a clinical setting. Finally, there has been some work using independent component analysis, or ICA, for seizure localization. However, these methods inherit the same problems as inverse source deconvolution. Namely, they require careful human annotations of the seizure interval, and the components are not guaranteed to reflect the seizure onset zone. And even if they do, it can be very difficult to select what the appropriate components are from the data. So I'm now going to dive into our work, and here I've grouped the two sections together because the models build off of each other. And really the crux of our approach is to recognize that instead of doing a kind of an independent temporal classification, the propagation of seizure activity actually provides valuable information about the onset. And so I'm going to start by describing our core framework, which is really geared towards seizure detection, which is modeling the spatiotemporal propagation. And then I'll discuss how we extend this framework for cross-patient localization. So this work was done by my graduate student Jeff at Johns Hopkins, as well as our collaborator Emily Johnson in the Epilepsy Center at Johns Hopkins. So our method actually relies on a framework called probabilistic graphical models. And as shown on the slide, these models have two components. There's a latent template, which captures unobserved phenomenon that we're trying to model. So in our case, this latent template will capture this unknown spatiotemporal propagation of a seizure. Now, this latent structure is complemented by observed data, which in our case will be features of the EEG signals. And these are really noisy observations that provide some glimpse or information about what is going on in terms of the seizure propagation. Now, mathematically, these two layers are tied with probabilistic models or probability distributions, and that allows us to propagate information from the data observations back to the latent structure. So I'm going to start by describing the latent template, so our propagation model. So here on the slide, I've illustrated the channels that we're looking at, so the EEG channels, which are marked in circles. So this is the 1020 International System, which is the most common montage used for epilepsy monitoring in the hospital. And on top of this montage, you notice these blue lines, and this creates a graph of seizure propagation. And what we have done to create this graph is connected neighboring channels to allow for local spreading, which is a very commonly observed phenomenon, and contralateral channels to allow for quick jumps across the hemisphere, which is one of the major confounds in visual inspection of these EEG data. So our model itself starts with all of our channels in kind of a baseline or non-seizure state. And as we march on in time, at some point a channel will flip and manifest seizure activity or be considered a seizure channel. And this could occur anywhere across the scalp, and it could be at different locations across patients. Now, as time moves on, this seizure activity will spread according to the graph. And so we might at first observe very sort of nearby or neighboring spread, and then it can propagate farther at later time steps until it might generalize to the entire scalp. So if we think about the temporal dynamics, this is very reminiscent of a hidden Markov model, which captures this kind of propagation of hidden states. And so a single chain hidden Markov model is shown on the slide. And so it consists of two portions. There's a latent state X, which are shown in the white circles, and then an observed data Y, which are shown in the gray circles. Now, an HMM is characterized by essentially two distributions. There's first the emission likelihood, which is how does the data Y, how is it generated based on the hidden state X? and then there's a transition prior. So given a state, what is the likelihood of transitioning to other states? Now, with just these two pieces of information, this chain can march on in time indefinitely. And then once we actually have data, what we observe are all the y's. And so in order to infer the x's, we run what's known as the forward-backward algorithm, where messages or sort of information about these probability distributions are passed both forward and backward along the chain in order to infer the likely state configurations. So with this as a backdrop, let's go back to our propagation model. So in our case, every channel, so every circle, can be represented as an HMM, and since there are many electrode channels in our montage, we essentially have a collection of HMMs, or chains, and in addition, these blue graph or lines between them actually couple these chains in what's known as a coupled hidden Markov model. So mathematically, if we look at the joint distribution of all these hidden states and all of the observed data, so x and y, we have an EEG likelihood, so again, how all the data is generated from the hidden states, which factorizes as shown. And then we have the propagation dynamics for each channel based on the previous state of its neighbors. So I'm going to start by describing actually the propagation dynamics. And so in our case, each channel we consider as being tri-state. And so the three states are pre-seizure baseline, seizure activity, and post-seizure baseline. And really here, the coupling is influenced by the probability of transitioning through these states with regards to its neighbors. So just to sort of give you a high-level picture, if we look at the bottom head plot, this channel T5 is grayed out, and so that's our channel of interest. So in this particular configuration, all of its neighbors are in a baseline state, so they're all green. So we would expect a very low transition probability into a seizure state, which is indicated by the orange dot on the left-hand plot, sigmoid. Now, if, however, a few of its neighbors were in a seizure state, then all of a sudden we have a higher likelihood of transitioning into the seizure state for T5. And likewise, if all of its neighbors are in a seizure state, we have an even higher likelihood of transitioning. And these dynamics are mimicked when we go from seizure back to post-seizure baseline. So now moving on in our framework to the observed data, well, if we go back to this kind of latent observed Bayesian model overview, when we think about interpretability, so our latent structure is actually giving us the interpretability. So we're essentially mapping the propagation of seizure activity or the states of all of these channels over time. Now, the EEG data itself, as shown in one of the earlier slides, is very complex. It has non-stationary characteristics over time, and also the seizure signatures and baseline signatures can vary. So rather than formulate a parametric likelihood, sort of a Gaussian mixture model or equivalent, we're actually going to use deep learning, and we're going to assume our likelihood is implicitly given to us by a deep neural network, in this case a convolutional neural network. And this is a model that we presented at IPME in 2019. So in this particular base model, we're going to train one CNN per channel as the likelihood, and this captures varying signal characteristics across the scalp. Now, if we kind of zoom in on this likelihood, we notice that there is a little bit of a problem. So by default, a CNN is a posterior inference engine. So if you think about a classification network, essentially what it does is it takes in data, so in our case Y, and it outputs a label, which is really like a posterior of the class given the data. But if we want to use this CNN as a likelihood model in a probabilistic graphical model, what we really want is the flip of that. We want a likelihood, so the probability of Y given X. And what we can do to obtain this factor is just rescale the CNN output using Bayes' rule. So the likelihood that we want, which is Py given X, it can be rewritten as the sort of quotient, and in the numerator, there's the CNN output, so the posterior, times the probability of the EEG data, so PY, divided by the seizure prior. Now, in terms of inference, this PY term is actually constant regardless of what state the channel is in, whether it's pre-seizure baseline, seizure, or post-seizure baseline. So for that reason, we can actually drop this factor and just consider the ratio of the CNN output to the seizure prior. And in fact, we can approximate this seizure prior PX with an empirical distribution. So across the training data set, we can compute the likelihood of a particular EEG window as being seizure versus baseline and pre-compute these PX values. Now, once we have this likelihood, we essentially can take our joint distribution and run a maximum likelihood optimization framework, so a variational or belief propagation, and obtain the full hidden state configuration. So we validate this model on EEG data from two different sites. The first is clinical EEG acquired at the Johns Hopkins University Epilepsy Monitoring Unit. And here we have 15 patients with focal epilepsy and 90 seizure recordings. So these patients had multiple seizures while in the hospital, and for each seizure we have between 5 and 10 minutes of baseline pre and post. The second data set is a publicly available data from Boston Children's Hospital. It consists of 24 pediatric patients with 185 seizure recordings. And here we don't have a characterization of whether they are focal or generalized epilepsy patients, so they could be either. And we compare our model with the same coupled hidden Markov model prior, but a parametric likelihood based on Gaussian mixture models, as well as just a pure deep learning CNN trained on signal channels. And so if we look at the quantitative performance of seizure detection, what we've plotted are AUC, F1, true positive, false positive, and precision, we see that in almost all cases, our method actually outperforms the baselines, which suggests that this fusion of the sort of probabilistic graphical model prior with a deep learning likelihood is providing gains over using each component separately. And beyond this detection performance, one really interesting and nice property of our model is that because we're tracking this spatial propagation, it allows us a glimpse at what we can do for localization. And so what I've shown here are just two seizure recordings for two different patients. And one is a right temporal seizure, the other is a left frontal seizure. And what you have are the red areas correspond to the posterior of our model, our combined hybrid model over time. And as you notice, our model, without ever having seen these recordings before, is able to infer the correct onset locations for each of these cases. And so this gives us kind of a starting point at which we can now take the core model and start to focus on localization. And to do this, we're actually going to build off of our hierarchical structure into what we call a regime switching Markov model. And this was work presented at McKay last year in 2019. And so from this larger figure, we notice essentially there's two components. The first is the core propagation model is going to allow us to track the spread and really provides that spatial resolution we care about in terms of localization. But what we are going to do is augment it with essentially a meta chain or a seizure detection chain, which really just detects at every point in time is the patient having a seizure or not. And the benefit of decoupling or this detection and localization problem, there's three main benefits. So the first is we can use multi-channel data to improve the seizure detection. So instead of training a CNN for each individual channel, if all we care about is detection, we can actually train deep networks on the essentially full data set across the scale, and that'll help improve performance. The second really critical benefit is that, as I mentioned earlier, these patients are sitting in the hospital and they're having multiple seizures, and each seizure provides information. So having this decoupling between detection and localization helps us actually fuse the information across all of these seizures into sort of a consistent statistical map. And then finally, we can isolate localization as a separate random variable, whereas in the kind of head plots or topological plots I'd shown on the previous slide, what we were essentially doing is just visually backtracking where the seizure started. But here we can actually hard-code it in and compute priors and statistics on that variable itself. So in order to expand this model, I'm going to move to a graphical model representation, which is shown on the right of the slide. And so here we have two plates, and that denotes conditional independences. So the inner plate are seizure recordings, so these are just seizure presentations from the same patient, and the outer plate is across patients, and so this is how we can learn across all our data set. Now, in this representation, we have squares and circles. So by convention, the squares are non-random but unknown model parameters, and the circles are random variables. And also, similar to the HMM diagram, the white circles are hidden, so these are our latent variables, whereas the shaded circles are observed. So I'm not going to go very much in depth with the mathematics, but just to give you a high-level overview of what each variable is. So this meta chain S is that top chain, the seizure detection chain, that provides a state for the patient, and it progresses through 5 states. So there's pre-seizure baseline, there's the seizure onset, there is the spreading of the seizures, so the propagation, the offset, and then post-seizure baseline. And essentially here, this F is really the EEG data concatenated across all channels. And so we can look at sort of deeper CNNs or multi-channel CNNs. We can also combine those with recurrent architectures, because really what we care about is an overall state. Once we have our seizure chain defined, within the probabilistic interactions, so X again defines the state, so pre-seizure baseline, seizure, post-seizure baseline for each individual channel. And here, the transition of S from baseline to onset will initiate the spreading pattern, which we discussed in our core model. And the transition from offset back to post-seizure baseline just resets the channels so that it might be able to detect another seizure coming up. And finally, this localization variable L indicates the onset channel for recording J in patient N. And the reason we have L as being a separate random variable for each recording is that, again, epilepsy is very heterogeneous, and so many times patients can actually manifest different seizure types, and patients can be multifocal, meaning that different seizures actually originate from different areas of the scalp. And we want our model to be able to capture these subtleties. And then finally, the way that we get a sort of a fused estimate for a patient is through that prior on L. And so across all recordings, pi gives us the statistical characteristics of the likely onset zones. Now, because of this 3-layer coupling, our inference procedure is a little bit more complicated. We actually use loopy belief propagation. So essentially, there's messages or information flow from the localization variable through the chains, through the meta chains. And so the pseudocode for our inference is shown on the slide. So we initialize our variables. We pass the localization information to the channel-wise detectors, which then gets passed to the meta chain. We run a forward-backwards inference on the meta chain. Then that information is passed to the individual channels, and we run forward-backwards through a variational approximation on the individual channels, and then we update the localization and then loop through this entire cycle. Now, we've actually applied this meta chain model, or this regime switching model, to the 15 patients from the JHU dataset where they have focal epilepsy, and we have a clinician who annotated where they believe the onset was coming from. And here are the results. So what I've plotted here is the variable pi, so essentially the prior over the localizations across all seizures. And the way we had run this experiment is that we essentially trained our model, so trained all of the likelihoods and ran our inference on 14 of the patients, so all of the seizures associated with those 14, and then we assessed it on all of the seizures associated with the held out patient. So this is interpatient or cross-patient localization. And so this experimental setup gives us an idea of how the algorithm would behave in a clinical setting where we would have our model calibrated ahead of time, and then new patients would come in, and we would have to on the fly make assessments about that new patient. And so in this particular figure, one thing we note is that essentially the prior pi has multiple modes in some patients, and so it can capture complex characteristics. These multiple modes can also indicate uncertainty, so if it was uncertain or if one of the seizures presentations indicated something different, then it would be reflected in this pi. And so just to parse these results, in the top row, we have illustrated the patients for which the primary or only mode of our model actually overlapped with the expert assessment, and that happened in 6 out of the 15 patients. And then in another 5 patients, what we have is our model had multiple modes, and the secondary mode actually agreed with the expert assessment. And so what's nice here is that we see in 11 out of the 15 patients, we have high concordance with the expert, so it has value for clinical translation. And we also have confidence in terms of having single or multiple modes. And so in cases in the top row, a few of the patients we identified a secondary mode that might be a trigger for an expert to go back and reassess their patient history, whether it's their EEG or other types of data, just to double check to make sure that the localization was correct. And likewise, if we disagree with the expert, again, it's a trigger for the clinician to go back and reevaluate. So now I'm going to wrap up with a nod to the future, and then I'm happy to take any questions. First, a plug for a recent tool that we've developed by a fantastic undergraduate, Danielle, in my lab. And so what we developed is an open source EEG visualizer. And so this tool allows you to load EDF files to plot the EEG signals. It also allows you to load Python modules, and especially PyTorch modules, and run different algorithms right on the data itself, and then plot the predictions. And so what we've just shown here are predictions for a particular model for seizure detection, but essentially this same tool can be used if you want to predict other signal characteristics like sleep awake. So it allows you to load the model and evaluate on the data. It also allows you to view annotations by the clinician as well as add your own annotations, to save and export the data, as well as to look at spectral characteristics. And so this can be downloaded from our lab website, and we hope it's a valuable resource to the community. So where are we going with this project? So essentially, our long-term goal is to develop an automated platform for non-invasive SOZ localization. And this non-invasive localization allows clinicians to plan more safe invasive monitoring and surgical resections. So in the first stage, which is what I presented, was to look at EEG. And so of course we're going to sort of improve our model, improve our training strategies, potentially add other types of clinical information. The second stage focuses on neuroimaging data, which is something that I didn't discuss in this talk, but it's also valuable in pre-surgical planning for epilepsy. And so we've done previous work on resting state fMRI for seizure localization, And so now we want to combine that with diffusion MR as well as structural signatures. And then the third piece of information is behavioral signatures during a seizure. These are often known as semiology. And so the semiology, it usually indicates certain types of seizures and certain seizure characteristics, and it can be used to influence spatial priors in both the EEG and the MR. And so our goal is to really fuse all that information together to provide a valuable clinical tool that works seamlessly in the current workflow. And before wrapping up, I'd just like to acknowledge my wonderful students, collaborators, and our funding sources. So in the personnel, everyone who's underlined contributed to the work in this presentation. So just to conclude, I've presented this hybrid of probabilistic graphical modeling and deep learning that allows us to mine multi-channel EEG data for seizure detection and localization. And so our approach is unlike anything that's been developed in the literature, and what we're able to show is that it provides robustness across our heterogeneous cohort, and it's also, to our knowledge, the first approach that can do the simultaneous detection and localization for multi-channel scalp. So with that, I'm going to conclude the presentation, and I'm happy to answer any questions that you may have. Thank you.